This method can help in the development of Reniform nematode-resistant cotton varieties through the identification of resistant genotypes from other cotton species and the evaluation of breeding populations. The main advantage of this technique is that it provides a simple and non-destructive method for Reniform nematode screening.
Begin by placing a ball of cotton in the bottom of one 4 cm diameter, 21 cm height conical plastic pot per seed, and filling the pots with steam pasteurized soil mixture to approximately 2 cm from the top of the pot. Place a plastic stake into each pot designating the genotype to be planted, and plant one seed of the appropriate corresponding cotton genotype into each pot. Fill each pot with additional soil to cover the seed, and place the pots in a growth chamber set to a constant temperature of 28 degrees Celsius with a 16-hour fluorescent and incandescent lamp photo period. Then place water emitters into each pot, and use an automatic watering system to water the pots twice daily. 7 days after planting, create a small depression in the soil next to each plant, and add 1 milliliter of Reniform nematode suspension into each depression.
After inoculation, pot watering needs to be carefully controlled in order to prevent washing the nematodes from the root zone. "28 days after inoculation, use scissors to remove most of the fully expanded leaves from the plants before squeezing each pot and sliding the soil out into one hand to remove the plants. Gently agitate.